is the Blackboard says that the homework assignment for lab was due yesterday. Just make it tomorrow like I told you. Sometimes I forget, and I apologize. The second thing is for homework for this coming Friday. It's an individual journal, so this is an individual assignment. All you have to do is just click on it. It'll take you to the prompt, and then you just click on Create Journal Entry, and you can begin to include your response. So this one is just a couple of questions. First, typical concentration of sodium chloride in its cytoplasm of a cell is 0.9. What happens to a cell if it's placed in a solution of 0.1% NaCl? And then how do gram-positives versus gram-negatives respond to this? So this is kind of taking us into what we're going to spend our time on Friday discussing, which is we're kind of going to go back to the phospholipid bilayer and talk a little bit about movement of molecules across the membrane. And then the second one is just a quick check. So we're almost 2 weeks through already. So are you keeping up? And do you have any questions, concerns, or etc.? So I'm the only one that sees these, and so that'll be due tomorrow night at 11:59. And then we'll use that as the first part as the start of our discussion on Friday. Tomorrow in lab, we are going to be gram staining again, and this time what we're going to be gram staining are the unknowns that you're going to work on for the next 4 to 5 weeks. Yeah, that's about right, somewhere in that time frame. So we'll talk more about that unknown project tomorrow and what ultimately the product for that project is. But tomorrow's main focus again is just gram staining and to confirm the identity, the gram status and morphology of your unknown. And I think that's all I have right now. 2 quick notes on things that I posted this morning in announcements. So if you have done that extra credit, which is the MHSCI quiz, test, survey, whatever you want to call it, you can just email me the 4-digit code that you receive at the end of it. And then the second thing is review session this afternoon, 4 to 5 in Predalin 306. So Julia will be there at 4 o'clock for an hour to help you out. Questions? Concerns? Okay. Let's go ahead and get started. And to start today, I'd like us to work on a case study. So rather than me sit here and talk for the next few minutes, what I'm going to do is I'm going to post this case study here for you to look at. Is that too small? I have a feeling it is. Let's just, there we go. Is that readable now? Better? I'll read it too, at least this first part. So it says, a woman comes to the hospital emergency room, room complaining of severe headache, nausea, vomiting, and pain in her legs. Upon examination, cerebral spinal fluid was observed leaking from a previous central nervous system surgical site. Patient indicates that 6 weeks and 8 weeks ago she had undergone CNS surgery after complaining of a migraine headache and sinusitis. Both surgeries involved a spinal tap. In the clinical microbiology lab, analysis of the cultures prepared from the CSF indicated no bacterial growth. Patient was taken to surgery where a large amount of CSF was removed from underneath the old incision site. The pinkish hazy fluid indicated bacterial meningitis. So among the laboratory tests ordered was a gram stain. Patient was placed on antibiotic therapy consisting of vancomycin and cefotaxime. CML findings from the gram stain CSF smear showed a few gram-positive spherical bacterial cells that often appeared in pairs. The results suggested Streptococcus pneumoniae infection. However, upon re-examination, a few gram-negative spheres were observed. And when transferred to a blood agar plate, which is a different type of growth media than what we've worked with so far this semester, growth occurred and the prepared smear showed many gram-negative spheres. Further research indicated that several genera of gram-negative bacteria, including Acinetobacter, can appear gram-positive due to under-decolorization during the alcohol wash step. Although complicated by the under-decolorization outcome, the final diagnosis was bacterial meningitis due to Acinetobacter baumannii. And so there are 3 questions below. From the gram-stained smear, what color were the gram-positive bacterial spheres? After re-examination, assess the reliability, and then what reagent is used for the decolorization. Okay? So take a couple of minutes, just discuss these over with a partner. This is kind of starting to tie together what we're talking about in class with what we've done in lab. So why does crystal violet only bind to the peptidoglycan of gram-positive bacteria? Allison?
Because of the outer membrane.
Good. What are the properties of a membrane? So we've talked about phospholipid structure. What are the properties of a phospholipid? This circle represents a phosphate group. Phosphates are polar, right? They're hydrophilic. What about the tails? They're hydrophobic. What did we say are the qualities of crystal violet? It's charged, it's got a positive charge on it, and it's a pretty large molecule. Those 2 features make it impermeable to the outer membrane. So because of not only its size, but because of the fact it has a charge, it can't actually get through that phospholipid bilayer. Okay? That's why crystal violet only binds to the gram-positive peptidoglycan, but not to the gram-negative peptidoglycan. Now, you may or may not be saying, wait, didn't we stain all different types of cells on Thursday with crystal violet? And the answer is yes. And that's because since crystal violet has a positive charge on it, still allows it to interact through ionic bonding or electrostatic interaction with the outer membrane. But it will get removed in the destaining step. We'll come back to that in a minute. Iodine is what we call the mordant. And what the mordant does is basically take crystal violet and it helps to create these large, typically they're described as large complexes of crystal violet. So you can imagine that the crystal violet is kind of getting into these open spaces within the peptidoglycan. That can be rinsed away with alcohol pretty easily. But what the iodine does is it starts to create larger complexes and basically locks the crystal violet into place within the peptidoglycan. So you can kind of think of the iodine here as a linker, taking 2 molecules of crystal violet and somehow linking them together so that they become locked within this thick mesh of the peptidoglycan. Next up is decolorization. What does the alcohol do? And Taylor, you started discussing one of the features. Can you bring that one up again?
It kind of dehydrates the bacteria.
Yeah, it dehydrates the bacteria. It starts to shrink. And in particular, it starts to dehydrate that peptidoglycan. So it's going to take this thick peptidoglycan and as it dehydrates it, kind of bring it closer together. The benefit of that is that helps to lock in the crystal violet even more. So at the point at which we're adding decolorizer, that crystal violet is permanently within the membrane, or within the peptidoglycan of those gram-positive bacteria. What does the alcohol do to a gram-negative? obviously we know the we observe that there's a washing away of the crystal violet. Why? What contributes to that? There's two answers to this one. First one is the crystal violet it's not locked in anywhere. It's just interacting through ionic bonds with this outer membrane. Second feature to think about is the properties of ethanol. Okay? How would we describe ethanol as a hydrophilic or a hydrophobic molecule? Does it mix with water? Yeah. Does it mix with oils? Actually has both properties. It's a little bit hydrophilic, it's a little bit hydrophobic. It can dissolve fats. Alcohols like ethanol and acetone too are able to dissolve or to wash away this outer membrane. So you can imagine that any of the crystal violet that's bound to it, when we add alcohol or ethanol in this case, it's going to not only wash away that outer membrane, but it washes away the crystal violet with it. And what those gram-negative cells are left behind with is now an inner membrane, a thin layer of peptidoglycan, and nothing else. That outer membrane is completely gone. Once we add the counterstain, that safranin, what's it binding to then? On a gram-negative cell, what does that safranin bind to? What's the most likely target? What was that?
Cell wall?
Yeah, it's the cell wall. It's the peptidoglycan, exactly. So at that point, what we find is the safranin is going to bind to the remainder of the cell wall, may interact with the inner membrane as well, but that's what gives us that pink color at that point. If we treat it with ethanol a second time, that safranin would be washed away pretty darn easily. Questions on that process? Why gram-positives are purple, why gram-negatives are pink? Annie?
So would the safranin also stain the gram-positive?
It does, actually. So safranin would also stain gram-positive, but why don't we see it?
Because the purple's already there?
Yeah, the purple's too dark. You're just making purple and making it more purple. Yeah. Other questions? Okay. Some of you may have answered this one as part of the homework assignment, but I'm just going to move ahead to acid-fast real quick. So here's an acid-fast bacterium. Inner membrane, peptidoglycan, not truly an outer membrane, but a waxy-like layer made of what we call mycolic acid, which gives the name to the bacteria that are considered acid-fast, the mycobacterium. Okay? These do not stain gram-positive or gram-negative. Why? I'll give you a minute to think about it. It has something to do with the structure of this envelope here. Okay? They don't stain positive or negative. Why are these unable to retain stain? How about this? Envision a candle. Okay. Candle is made out of what? Wax. When you add a drop of water to wax, what happens? Yeah, exactly. It kind of balls up, right? If you were to add food coloring to that, does the wax become stained? You should try this at home. Okay. The wax doesn't become stained. What would we have to do to actually stain the wax? if we wanted to like change it from a white color to say an orange color, what would we have to do? Think of this another way. Maybe you want to stain a stick of butter purple. What would you have to do to that butter to stain it purple? Exactly, you've got to melt it. Why? The wax is a solid, essentially, right? So because of the waxy-like coat that we find on Mycobacterium, they're resistant to retaining stain. The stains that we use are charged. Safranin and crystal violet are both hydrophilic molecules, and so they don't mix well with the wax. The wax being solid is not going to retain it or hold on to that color overall. In order to be able to stain it, we have to actually melt that wax somehow, or that mycolic acid. Acid-fast bacteria, if we were to do traditional gram staining, would appear blank. We would not see anything at the end of the process. To stain acid-fast bacteria, there's a slightly different staining technique, which is known as the acid-fast technique. First, heat these up. So you take a slide with your active fast bacteria and you heat it over boiling water, basically on like a bed of steam. That helps to melt the mycolic acid. Then you add a dye. After adding that dye, you snap chill them. You cool them down very rapidly. And what does that do? Well, it rehardens the wax, and it allows for any of the dye that was present that made it past that wax layer to now be locked into the cell. The dye we typically use for this is known as carbolfuchsin. It's a reddish color, and so acid-fast bacteria through this staining procedure would typically appear red once we're done with them. Okay. Prototypical members of the acid-fast, as I mentioned, include the Mycobacterium. Tuberculosis is a bacterium that has this type of waxy surface. And this is a really good way to avoid immune detection too. It's kind of similar to the glycocalyx that we talked about on Monday. Annie?
So you're actually staining the mycolic acid and not the...
Yeah, it's kind of a combination here. A little bit of the stain will stay in the mycolic acid, but also by melting it, you're allowing for that stain to kind of get past the mycolic acid and almost become like trapped inside of the cell, or in between the cell membrane and the mycolic acid as well. Okay, I think that's all we needed to cover with acid-fast for right now. So 3 different types: positives, negatives for gram staining, and then the acid-fast bacteria. Each of them have slightly different characteristics. Each of them have features that make that help us to identify them. Okay, not only based on phenotype of the type of staining, but also then what's happening at the cellular level that contributes to some of those differences in staining as well. The other thing that I wanted to point out was in the table it asks about sporulation. So sporulation is the ability of a microorganism to form what we call a spore. Spores are a lot like seeds are to plants. Okay, so end of the season, a seed gets formed as conditions become harsh. It's a way to ensure that genetic information gets passed on from one generation to the next generation. And a seed can remain dormant for years. Okay? When the conditions are ripe, what do we need for a seed to sprout? I know everybody has done this in at least 2nd grade or 3rd grade, where they gave you a bean and they said, grow a plant out of this. What do you need to do? You do not have to have taken botany here to answer this question, surprisingly. What do you need to make a plant grow?
Water.
Water. What else?
Sunlight.
Sunlight. What else? Soil, nutrients, carbon dioxide. Okay? The conditions for a seed to sprout, they have to be right. If you don't have any of those, you lack the ability for the seed to actually grow. Same is true for a spore. Not saying that it needs those same nutrients and sunlight, etc., but for a spore to be able to grow, the conditions have to be correct. Often involving nutrients, heat, to get the cell growing. And what we find with spores is, we'll discuss the structure of these a little bit later. I'm trying to remember when exactly. I think it's next week we'll discuss them. Yeah, next week. But spores have a really thick coat, kind of like the shell of a seed, that help to protect the genomic material inside. We, in laboratory conditions, there are spores that are 30,000 years old that have been germinated in laboratory conditions. So they are incredibly hardy and incredibly resistant to environmental conditions. Where we see these in medicine and hospital settings, Clostridium is one of, there are a couple of different types of genus of bacteria that are capable of sporulating. It's limited to gram-positive, and it's limited to Bacillus gram-positive. Only a few genera of Bacillus. So Bacillus subtilis, Bacillus megaterium, Bacillus anthracis, which is the cause of what? It's in the name, anthracis. Anthrax, okay, are all sporulating bacteria. Clostridium, on the other hand, is another. Clostridium botulinum, Clostridium tetani, Clostridium difficile. I'm sure everybody's heard of all three of these, right? Tetani causes tetanus, botulinum causes botulism, difficile, C. diff, exactly. The one that people most often, when I bring that up, the faces kind of go goofy because I've never experienced, but I hear it smells absolutely awful, okay? And that's the one that in hospital settings you will most often deal with. One of the things that we have to talk about later on in the semester is what makes these spores so hardy and how do we actually get rid of them. So that becomes a conversation for a later point. But just know that they do exist. Okay, we've actually got enough time to finish up with a couple of questions today, but first I want to see, are there any additional questions on what we've covered?
This is kind of off topic.
That's okay.
But with C. diff,
Yeah.
with sporulation, I mean, I don't know, I've heard like from doing clinicals and stuff that C. diff can be caused because of like the amount of drugs you're on.
Oh yeah.
Like with, does that have anything to do with?
Yes, and we will talk about this. And when they say drugs, they usually mean antibiotics.
Yeah.
So C. diff is what we would describe as an opportunistic pathogen. And what we mean by that is that the individual is immunocompromised in some ways. We have found over the past 20, 30 years, too, that immunocompromisation or being immunocompromised doesn't only mean that your immune system has some sensitivity or issues. It also means the natural bacteria that are inside of you have a defect as well. So one of the things that we'll talk about later on in the semester is when you take an antibiotic, an oral antibiotic, you're not only killing the microorganism or inhibiting the growth of the bacteria you want to get rid of, you're also possibly inhibiting the growth of any other bacteria that come into contact with that drug. Doing that frees up space. I like to think of it as like you have a city full of people and then you ask everybody to move out and then like you have stragglers move in. That's C. diff in this case, where all the bacteria, I shouldn't say all, majority of the bacteria in the gut are killed or are inhibited in their growth. That creates room for C. diff to colonize. So that's why we describe it as an opportunistic infection. Okay? We'll talk about that later. That becomes, honestly, third unit of this semester is focused on mutations, antibiotic resistance, and some of those other features. And we'll come back and actually talk about that one, even right around spring break. We'll talk about that one as well. It's a great conversation piece. Any other questions? All right, let's get talking. Lysozyme, enzyme that is found within tears, in the mouth, etc. Actually, before the invention or before the discovery of penicillin, Fleming, who discovered penicillin, was working with lysozyme as a potential antimicrobial, and it is a very good antimicrobial. Okay, so here lysozyme, what it does is it hydrolyzes the bonds between N-acetylglucosamine and N-acetylmuramic acid. What does hydrolyze mean again? If it drives a hydrolysis reaction.
Breaking apart.
Good, breaking apart. Water being used to help break this bond apart. So specifically, it's breaking this bond right here. So the question is, what would happen to a gram-positive cell that was incubated with lysozyme? We'll just stop there to start. Let's just start with what's going to happen to the cell wall. Okay? And then the follow-up question, which deviates from what's on here, what will happen to a gram-negative cell wall in the presence of lysozyme? Okay? I'll give you, let's work on this for like 5 or 6 minutes, and then we'll go ahead and talk about this one afterwards to finish up for the day. The bonds that occur between N-acetylglucosamine and N-acetylmuramic acid, what that's going to do is it's going to start to disturb or break apart that lateral integrity of the cell wall. Okay? Obviously a bad thing. Okay. What will happen to a gram-negative cell? So let's go back to our image of gram-negatives.
Will it be protected by the outer membrane?
Why?
Because it is.
I'll preface this as saying you're right. I want you to go a little bit deeper with it. So what are the properties of the phospholipid bilayer that would inhibit or prevent lysozyme from getting through the membrane? That's one of them, yeah. And without telling you ahead of time, lysozyme is a hydrophilic molecule. We find it in tears. We find it in mouth. It bathes itself in aqueous solution. Okay. What else is true? An enzyme. What is an enzyme? What kind of macromolecule is an enzyme? It's a protein. Proteins, big or small? Big. Much larger than a phospholipid bilayer. Look, we've got proteins. Here's an example of one I'm pouring. This is embedded in the membrane. A hydrophilic protein, will that be able to permeate or get through this cell membrane here? No. And so what we find with gram-negatives is that they are not affected by lysozyme treatment because of the presence of the outer membrane. Gram-positives, on the other hand, we observe breakdown of that cell wall. What does that mean then in a hypotonic solution? What does hypotonic mean? And this is kind of leading into Friday, but I know that this is a term probably some of you have heard.
We pull the fluid out of the cell?
Yeah, other way around. So if we're saying that a solution is hypotonic relative to the cell, that means there's less solute outside than inside, and so the water movement would be into the cell. So typically, when we add water to a plant cell, for example, right, what happens to that plant cell? What's that? It increases in size, right? It swells. That's why, like, some people like to store carrots or celery in water because they stay nice and crisp, right? And we would see this swelling occurring for a bacterial cell as well. That's going to increase in size. And usually, that peptidoglycan is there to prevent further expansion. If lysozyme's added, what's going to happen to the cell, to a gram-positive? They're going to lyse, they're going to break it apart, or they're going to explode. Because by breaking down that lysozyme, or that outer layer, what that's going to do is it can't control or prevent the continuous turgor pressure that's being put on the cell by water moving into it. Okay. this kind of becomes a part of our conversation on Friday, but why go through a problem like this one? Couple of reasons. One, it's a darn great review question, because what does it combine? It combines what we've talked about in chapter 1, a hydrolysis reaction. We talked about, hey, enzymes are macromolecules, and I could follow that up with, it's a protein, and proteins are made up of amino acids. So it covers that first chapter. It covers structural differences between gram-positive and gram-negative cells. It talks about the purpose of the cell wall. And it gets into what we're going into on Friday with homeostasis across the bacterial membrane. This is an old exam question. Okay, so if you are wanting to know what's the first exam going to look like in, say, 16 days, which is the actual number, this is what you can expect to see. Not throughout the whole thing, okay, but at least roughly half of the points are made up of questions like this one. So that's why I'm trying to stress now, start practicing with this. These are the review questions that I put up for you. Start practicing with them, okay? All right, that's all I've got for today, so I will see you tomorrow. I do have quizzes from yesterday, so pick them up on your way out, and if you got questions, we'll have plenty of time tomorrow, so you can ask them too.